then we'll do the fun activity, then we'll get out of here, you'll study, and then next week I crush your dreams. All right, so.
No, come on.
Did you just say crush your dreams?
Yeah, it's all, I got it all planned out. That's the final step. All right.
We're talking this way, yeah.
Okay, so let's see, where were we on this thing? We got down, what do we?
I think we did all the heart. Yeah, we're at chapter 21.
All right, chapter 21, cool. All right, so it says matching category with the following choices: artery, vein, capillary, arterial, venule, all of the above, none of these, and that comes up in this next section. This thing is so redundant in how they, I didn't write, I not only did I not write the final exam, I did not write the final exam study guide. And I would love to change it, but I can't because we're all supposed to use the same one, so whatever. All right, differentiate between the type of blood vessels in terms of anatomy, functions, and location. So, by definition, what's an artery? An artery carries blood away from the heart, and what's a vein? Carries blood towards the heart, right? So let's go through like the general setup. As you leave the heart, you'll first go into arteries. What come after arteries? Arterioles, and then you go into capillaries, then you go into venules, then you go into veins, right? That's the general format there. Remember, for most of those blood vessels, there are going to be 3 layers to the wall of the blood vessel, right? What's the name of the inner layer? Yeah, the tunica interna, or intima is the inner layer. What's the middle layer? Tunica media, and then the outer layer is the tunica externa, right? The only exception to that are capillaries, right? Capillaries usually only have 1 layer. They'll have that. They'll have the tunica interna. All of your blood vessels. What's the type of epithelium that you have in all of your blood vessels? Simple squamous, right? Which is the thinnest type of epithelium possible. If you compare arteries with veins, you'll find a thicker, smooth muscle layer in arteries than in veins. Why is that?
Because of the constriction of it?
Because they carry higher pressures. Remember, the arterial side of things is high pressure. The venal side, the vein side of things is low pressure, so you need to have thicker walls. Another thing about arteries that I'm pretty sure I didn't mention is they have something called an internal and external elastic membrane. These are little membranes that are kind of sandwiched in between the tunica interna and the tunica media, and then between the tunica media and the tunica externa. That's something you just see in arteries. Again, they're called internal and external elastic membranes. Not all arteries have them. It's usually just like your larger arteries have them. Veins, what can I say about veins? Which do you have more of, arteries or veins? You have more veins, right? Especially in your extremities. Remember, you had a deep set of arteries in your arms and your legs, and that's it. But for veins, you have a deep set of veins, and then you also have a superficial set of veins in your arms and legs. So there's more veins than there are arteries. And at any given time, there's more of your blood in your veins than in your arteries. So they sometimes refer to the veins as blood reservoirs. Okay. Now the problem with veins, again, is that there's low pressures in the veins, and that makes it hard to keep the blood flowing. What are the 3 things that keep blood flowing through the veins? Skeletal muscle pump, respiratory pump, and the valve. Sorry, I was going to give a clue, and I just like gave you valves. That's it. It's going to get too stupid clue. All right, that's right.
Those are the autonomic.
What color are arteries usually? Red. And what color are veins usually? What were the exceptions? The umbilical ones, and then also what were the other?
Hepatic.
The pulmonary, right?
Okay.
So the umbilical and the pulmonary were exceptions. Umbilical arteries are blue, umbilical vein is red, pulmonary arteries are blue, and pulmonary veins are red, right? Where is it going with this? Capillaries are where the exchange happens, right? Capillaries have a thin layer. They only have that inner layer. They only have the simple squamous epithelium. That's to make it as thin as possible so you can exchange gases and other materials there. Remember, when you do capillary exchange, there's going to be some amount of fluid that leaves at the beginning of the capillary bed. But by the time you get to the end of the capillary bed, what's happened to most of that fluid? It's come back in, right? Remember, when the fluid leaves at like the arterial end of the capillary bed, that's called filtration. And then when it comes back in at the venule end, it's called reabsorption. What percentage of the fluid didn't come back?
15.
About 15%, and who picks up that fluid?
Lymph.
Lymphatic system, right? Remember, there are 4 pressures that govern that sort of capillary exchange. 2 of them are trying to push the fluid out of the blood and into the tissues. That's the blood hydrostatic pressure and the interstitial fluid colloid osmotic pressure. And then 2 of the pressures are fighting against that and trying to push the fluid from the tissues back into the capillaries, and that would be the interstitial fluid hydrostatic pressure and the blood colloid osmotic pressure. Of those 4 pressures, the only one that really changes from the arterial end to the venule end is the blood hydrostatic pressure. But that makes a big difference, because if you do that kind of math problem of net filtration pressure, anyone remember the value that you got at the arterial end when you did the math problem? It was like 10 millimeters of mercury. The fact that it's positive means that fluid is leaving the capillary and going into the tissues, right? What if you do the same thing at the venule end? What value did you get? It was negative 9, okay? The fact that it's negative tells you that it's being brought back in. But because it's not exactly opposite, because it's not negative 10, that implies that some of that fluid that left isn't coming back in, and that's why you got to rely on the lymphatic system to pick it up. All right,
That was just the first bullet.
what's that?
Mm-hmm. That was just the first bullet, right?
Yeah, I'm still just on that first bullet. There's a lot to say.
Right.
Yeah, sorry, there's a lot to say.
No,
Well,
it's fine.
if you remember from when I laid out the questions during last class, this is also a chapter that has a lot of
Yeah.
questions, like 19 are from this chapter. So,
Okay.
arterials, so the usual pattern is to go artery, arterial, capillary, venule, vein. What was the one situation when you went from arterial to capillary to arterial again?
Glomerulus.
The glomerulus, right? You go from the afferent arterial to the capillary to the efferent arterial, right? By the way, what kind of a capillary did you have in the glomerulus? Fenestrated, right, with the little holes inside of it. All right. Do blood vessels receive parasympathetic innervation? Not usually. Usually they're just connected up to your sympathetic division. So if you want to control how much vasoconstriction or vasodilation there is, you're usually just adjusting how much sympathetic signaling you're sending. It's usually not the parasympathetic that gets involved with that. Matching category with choices. Increases blood pressure, decreases blood pressure. So let me throw some random things out, and you tell me whether you think this would make the blood pressure go up or go down. Aldosterone. What's the correct answer? Up. So aldosterone will make your blood pressure go up. Atrial natriuretic peptide. That'll make your blood pressure go down. ADH. Up. That makes your blood pressure go up. Wow, we're really crushing this. How about increased cardiac output? Does that make your blood pressure go up or down? Up. Okay. Decreased cardiac output makes your blood pressure go down. Okay. Here's a formula that you might want to try to remember for figuring out blood pressure. Okay. Remember, this was a rearrangement of Ohm's law, and it's basically saying that the mean arterial pressure, which again you can just consider that your blood pressure, because that's usually what we're talking about is your arterial blood pressure. So your blood pressure equals the cardiac output multiplied by the systemic vascular resistance. Okay, so anything that makes the cardiac output go up or anything that makes the resistance go up is going to make the pressure go up. So that may help you to answer these questions, or maybe not, but anyway. So if cardiac output goes up, pressure goes up. If cardiac output goes down, pressure goes down. If you increase the resistance throughout the body, that'll make the pressure go up. If you decrease the resistance throughout the body, that'll make the pressure go down. Some other little tidbits. Norepinephrine, this is crazy, but this comes up on the final. Norepinephrine on alpha-1 receptors of blood vessels. So on the smooth muscle that's in the walls of the blood vessels, there's a type of norepinephrine receptor called an alpha-1 receptor. And what they're asking you is, what happens to your blood pressure when norepinephrine stimulates the alpha-1 receptors? And what do you think? You've got a 50. First of all, let's just think about this. Norepinephrine is released by the sympathetic division during fight or flight situations. So what do you think that would do to your blood pressure?
Increase it.
Makes it go up. Okay. So norepinephrine on alpha-1 receptors makes your blood pressure go up. Histamine. What does histamine do to your blood pressure? What is histamine? It promotes what?
Inflammation.
Inflammation. So it usually makes your blood pressure go down. Okay, histamine, whether your body's releasing it from having an infection or it's an allergic reaction, histamine will tend to make your blood pressure go down. Here's one that's a little bit trickier. I'm going to have to walk you through this. What if you have a decreased solute concentration in the blood? So by that they mean, what if you had less protein, say, inside of your blood vessels? What would that do to your blood pressure? No one know? It has to do with osmosis. I'll give you a hint.
Was protein in or out?
There's less protein inside your plasma. What would that do to the blood pressure?
Decrease it?
It would make your blood pressure go down because there's less of a drawing force pulling water into the blood vessels. Okay? Just saying that.
I'm trying to visualize it, like.
Yeah, there's less if you have less protein is an impermeable solute, right? It's not going to go through the walls of the blood vessels. So if there's lower levels of protein, there's less of an osmotic draw to pull water into your blood vessels, which makes your blood pressure go down. Okay? I didn't really belabor that point personally. I didn't think it was a huge deal, but guess what? It's on the final, so I mentioned it. Okay. Good enough. Just remember this formula. This is a pretty important formula. Remember, there's little things that can feed into this formula. You can keep taking a step back from each of these things. So cardiac output, anyone remember the formula for cardiac output? Stroke volume multiplied by heart rate. So anything that increases the stroke volume or increases the heart rate would increase the cardiac output, which in turn would increase your blood pressure. Okay? You can keep taking steps back from that. You know, your sympathetic system, turning that on, increases the heart rate, which increases your cardiac output, which then increases your mean pressure. On the other side of things, the systemic vascular resistance, remember you can take a step back and say, well, what increases resistance? Well, if you vasoconstrict your blood vessels, if you tighten them up, that's one thing. That would increase that, which then would increase your pressure. What if you just suddenly made your blood thicker, like more viscous, right? That's going to increase the resistance, and that'll bring your pressure up. What if you just decided to get fat, right? That's a good option, right? That's always an option, right? You decided to get fat, then that created more blood vessels. Remember, more blood vessel length also increases the resistance, right? And that'll increase your pressure. So all of those things. All right, where am I going with this? Where am I? Okay, lymphatic system and immunity. Differentiate between non-specific and specific defense. First of all, what were the other names we had for non-specific? Innate or inborn, right? And then what were other names we had for specific?
Adaptive.
Like adaptive, or sometimes we call it acquired. So they're all names for the same thing. And remember, when we thought about immunity, we thought of it in terms of lines of defense. Which division provides the first two lines of defense? Yeah, non-specific or innate provides the first two lines of defense. Who provides the third line of defense?
Specific.
Adaptive or acquired, whatever you want to call it. So let's go through those lines of defense then. What is the very first line of defense? Essentially barriers, so the skin on the outside of your body or like the mucous membranes on the inside of your body, right? That's really like the best line of defense. Keep the stuff out of your body in the first place, right? But if you break through that, then you got to go to the second line of defense. What were some of the things that were in the second line of defense? So inflammation was there, fever was there. What kind of cells were involved in the second line of defense? Neutrophils, macrophages, natural killer cells, all of those guys were part of it, okay? So both of those parts, line 1 and line 2, were both part of innate immunity, right? If you break through then you have to go to the third line of defense, which is provided by adaptive immunity. And let me go through column. Sorry, I just gotta check this. Sorry. Okay. Guys, like, I knew this guy can't possibly miss this
Nah,
today. I was like,
nah.
Okay.
This day of all days I'll never miss. No.
All right.
That's
Um,
Tuesday.
Yeah.
You can't play because you're late.
I almost I almost brought my stash of Xanax in for you. I need it when I fly, and I got some trips planned, so I was like, I just can't part with it. I'm sorry. I wish I had some that I could.
No, I admit, since when I was talking to myself this morning, I was like, you know what? I'm gonna be calm. No, really, like whatever happens. Like,
That's great. I like that.
You know what I mean? Like,
He's ready for the Boston Knicks game tonight.
I think this is a new you. This is a turning point in your life. I think this is like, yeah.
That's why I came late.
Listen, I'm an optimist. I think you're gonna see.
Yeah, that's why I was late. I just revelation. It's like so hard. It's like.
All right. I can't wait to see the new you. I'm really pumped. All right. Moving right along. Where the heck was I? So adaptive immunity provides the third line of defense. What kind of cells should you think about when you hear about adaptive or specific immunity? Yeah, and B cells, right? So lymphocytes. Think lymphocytes. T cells and B cells, right? They're part of your adaptive immunity or your specific immunity, whatever you want to call it. Now, within the branch of specific immunity, there were two sub-branches to that. What are the two sub-branches of specific immunity? Cell-mediated immunity and antibody-mediated immunity, right? Cell-mediated immunity, you should think of what kinds of cells? Cytotoxic T cells, right? And for antibody-mediated immunity, you should think of what kind of cells? B cells. What's the name of the cells that kind of play a role in both branches?
Helper T
T
cells.
helper cells, right? They're going to play a role in both branches. All right. So let's talk about some of these things that we have here. What kind of problems, or what kind of infections, or whatever? What problems, infections, however you want to put it, is cell-mediated immunity good against? Yeah, usually intracellular infection. So that usually means viruses. There are some weird kind of fungi that they're good against. Cancer, it's usually cell-mediated immunity that's on the lookout for cancer cells. Unfortunately, cell-mediated immunity is also what causes rejection of what? Transplants. Now, I will tell you on the final, they don't use the word transplant, they use the word graft. It means this, I know. If you have any question, just come up and ask me. But it means the same thing as basically a transplant. So you can call it a graft or a transplant. It means the same thing. And then what kind of problems is antibody-mediated immunity good against? Bacteria, things that are usually what? Extracellular, okay? So bacteria, as well as the toxins that are released by bacteria, all that stuff is usually extracellular for the most part. There are some bacteria, for those of you that take in micro, like, tuberculosis, for example, are intracellular, but most bacteria are extracellular. All right, it also says co-stimulation, MHC used. So remember, to turn on your immune cells, you need a couple of things, and it depends on whether you're trying to turn on T cells or you're trying to turn on B cells. T cells are a pain in the butt. Remember, they needed to have the antigen shown to them. They needed the cell that's showing it to show them the foreign antigen, but they also had to show their own self-MHC markers, and you still needed something called co-stimulation, which is the second chemical to be released. If you had all that stuff, then maybe those lazy-ass T cells would get involved, right? You needed all of it. B cells were more chill. B cells can pretty much just kind of do it on their own. They don't need an antigen presenter. They can do it on their own. However, if they suck it up and just admit that they need help every now and then, and if they do get a helper T cell involved, it will make the process faster, than doing it alone. Now, what else to say? Okay, moving right along. Sorry. Describe dendritic cells, macrophages, self-tolerance, complement fixation, haptens, and natural killer cells. So anyone remember what dendritic cells were? Okay, what do they look like?
Branches.
They have a lot of branches, right? They look kind of like a neuron with the dendrite branches, right? They were classified as specialized antigen-presenting cells. So they are very similar to macrophages. They both are good at phagocytizing things and then presenting it to like T helper cells, okay? That's what they're specialized antigen presenting cells, okay? Remember, to turn on a T helper cell, you needed a specialized antigen presenting cell to first phagocytize the material and then to show it off on their surface in association with an MHC2 class molecule in order to turn on a T helper cell. Remember, for cytotoxic T cells, any infected body cell can serve as the antigen presenter, and instead of using MHC2, they use MHC1 to make the presentation. All right, self-tolerance, what does that mean? Does that mean not being hard on yourself after you fail this class? What does that mean?
Yes.
That part, yeah, it does mean that a little bit. No one's gonna fail. Wait, where's some, I need serious winning. All right.
Video.
No, what does self-tolerance mean? Bless you. Just basically means not like attacking your own body cells, right? Being able to recognize them as being a part of you and then not attack them. And a lot of that depends on being able to see the self-MHC markers, knowing it's a part of you, right? All right, complement fixation. Now, what's the complement system? I like your shoes, you like my hair. No, that's not the complement. What?
Starts something and then the other one can start something.
Was that your description? One thing starts something, then another thing starts something?
Means.
Okay, that's fine. No, I like that. You were so vague that you can't possibly be wrong with that answer. Yeah. So, what's going on with complement fixation? Remember, it's basically like coagulation. One chemical leads to another chemical leads to another chemical leads to another. And then what started off the complement system? C3 started it off, okay? And the three benefits that you got at the end after you turn it on were enhanced phagocytosis, enhanced inflammation, and cytolysis, making this thing called the membrane attack complex. Now, the term fixation is stupid, okay? But that's on the final. Complement fixation just means to turn on the complement system. Why they had to use the word fixation or fix, I don't know. Don't ask me, okay? When I first read it and they said fixing complement, I said I didn't know it was broken, okay? But anyway, it just means to turn on the system. If you have any questions during the final, come up and I'll remind you that's what complement fixation means. Haptens, I definitely didn't cover haptens in class. But if you read the textbook, did anyone accidentally read the textbook this term? You like trip across it and the page flew open and you accidentally read a word there or something? Just, no one read the textbook, good. I was worried that somebody actually read the textbook. So in the textbook, it actually explains what haptens are, but let me tell you what a hapten is. A hapten is kind of like an incomplete antigen. What I mean by that is, you know, when you put an antigen, when you put a full regular antigen into your body, it will trigger your immune response and you'll mount a response to it, and then you'll start to try to fight it off. An incomplete antigen, a hapten, is something that in and of itself, it won't trigger an immune response. It has to get into your body, number one, and then it has to attach to one of your own body proteins to sort of become a complete antigen that will then actually trigger your immune system. You're like, what the hell does that mean? Let me give you a very specific example. Anyone here ever get poison ivy?
Yeah, it's the worst.
Poison ivy, the proteins that are in poison ivy are considered haptens. In and of themselves, they are not able to trigger an immune response. But if they get into your body and they bind to special proteins, then they can trigger an immune response and you get poison ivy. Okay, that's
Some people like get it worse than others.
That's right. That's right. All right, so that's what a hapten is. And then natural killer cells. What kind of cells are natural killer cells? They are the same thing as what are B cells and T cells? They're lymphocytes. Natural killer cells are lymphocytes, but what sets them apart from like your regular T cells and B cells is that natural killer cells are a part of innate immunity, whereas the T cells and the B cells are part of adaptive immunity, right? All right. Describe the
7 questions.
So, what's that?
Sorry, I just thought out loud. Like that was 7 questions. Like that was.
Yeah, I know. There's there's only 7 questions from this, but yeah. By the way, just so you know, I give you pretty much everything you need to know, but I also give you more than you need to know. So
Thank you.
Overqualified.
Yeah, I feel like it's too. I feel too dirty. I feel too unseemly just feeding you answers. So I gotta feed you a whole bunch of
Take a
like
shower.
bullshit and the answers. All right? Yeah. It just seems. It doesn't seem right just giving you pure answers. I gotta mix in some crap with that. Okay? Yeah. Sorry, it just doesn't feel right giving you. All right. So 5 IgG classes. What can you say about IgG? It's the what? It is the most common. For those of you that don't like synonyms, on the final it uses the word abundant. It's the most abundant. That means the same thing as the most common. Whatever. Also, it's the only one that can cross the what? It's the only one that can cross the placenta. Okay. Also, to be aware that, and this is how it's worded on the final, it can fix complement, which just means it can turn on the complement system. Fine. Who wrote it? Is that what someone asked? I don't know who wrote it. Well, it was the whole science department a long time ago. Before I even got there, they wrote this thing.
It was Barbara,
Yeah.
wasn't it?
It's freaking ancient. Yeah, it's ancient.
She was a heckle, wasn't she?
This was back when they were driving to school with cars that had square wheels. I mean, it was a long time ago. Okay. Where am I? I'm sorry, I'm getting all distracted. All right, antibodies again. What's special about IgM? What does IgM look like?
Oh, is it
It looks like a snowflake, right? It's a pentamer. It's made up of like 5 of those parts to it. Also, too, what's special about the timing of IgM?
Right after.
It's the first to appear, right? But it's also the first to disappear, too. So if you see high levels, what does that mean? It means you're early on in your infection, right? Also be aware that it can, too, turn on or fix complement, okay, just like IgG can. IgA, what's special? What's the shape of IgA?
Dimer?
It's a dimer, and what's special about the location of IgA? Secretions, right? So it's in-body secretions. These other ones, IgM, IgG, they're mostly in your blood, but IgA is inside body secretions, breast milk, saliva, tears, all that stuff. Okay. And then IgE and IgD. Who cares? Who cares? All right, next. Describe the 4 types of immunity. So we had 2 parameters here. We had active versus passive. What are you being exposed to, or what are you being given with active immunity? The actual antigen, which will then trigger your immune system to respond to it. And because of that, you'll create memory cells. So you get long-lasting benefits. What are you being exposed to or given with passive immunity?
The antibody.
The antibodies directly, which are awesome while they're in your system. They'll fight, they'll take care of all kinds of problems, but they're kind of doing the job for your immune system. And so you don't ever trigger an actual immune response, so you don't create memory cells, which means with passive immunity, you don't get long-lasting benefits, okay? The other parameter is natural versus artificial. What does natural mean?
The environment.
You're just going about your day-to-day life, and you somehow acquire this immunity. What does artificial mean?
Given
You actually sought out like a healthcare provider, right? You've seen somebody and you got some kind of treatment. So let me just give you some specific examples here. So what's a specific example of artificial passive?
Okay.
So yeah, sure, Rogam could be an example of artificial passive. What else?
Snake venom?
Snake antivenom, right? That's a good example too. What's an example of natural passive? Like breastfeeding, right? With the breast milk, the baby getting the beneficial antibodies there. What's an example of artificial active?
Vaccine?
Vaccine. So like getting a vaccine against measles or smallpox or some disease like that. That's an example of artificial active. And then what's an example of natural active? Just getting infected with something, right? Okay. Good. Okay, moving right along. Where am I? I'll just go. Okay, lymph enters venous circulation in between what 2 vessels? So this is basically asking you, where does the thoracic duct and the right lymphatic duct empty into? It's basically the subclavian vein. The reason why they say between what 2 vessels is because if you look, the exact spot where they empty into is kind of at the junction of where the subclavian vein meets the internal jugular vein, okay? So just be aware of that. All right, 23, respiratory system. Describe the cells found in the alveoli in terms of their type and function. So first of all, what kind of an epithelium do you have in the alveoli? Simple squamous, which is the thinnest epithelium possible. What's the name of the alveolar cells that actually make up the simple squamous epithelium? They are called type 1 alveolar cells. Okay, they make up the wall. And obviously with a name like type 1, you probably surmise that there's type 2. What did type 2 do?
Does it help keep the lungs?
They make- Yes, you're both right. Yeah, so they make surfactant, which helps keep the lungs open when you exhale. They break the surface tension, which is this natural tendency for the moisture inside the alveoli to pull those alveoli and make them collapse every time you exhale, which would then mean when you want to take your next inhalation, you'd have to try to pop open all these closed alveoli, and that takes a lot of strength. Who are the individuals that don't make enough surfactant? Yeah, preemies. Premature babies don't make enough, and they have to be put on a mechanical ventilator to help them out. When inhalation occurs, is the diaphragm contracting or relaxing? It's contracting, right? And when the diaphragm contracts, which way does it move? Up or down?
Up. Down.
Down. So the diaphragm goes downward, and that makes your thoracic cavity get kind of bigger in this dimension, right? Also, at the same time, usually you'll have like the external intercostals that are between your ribs pulling out, so they're making the thoracic cavity bigger in this dimension. Okay. What's the name of the law that governs ventilation? Boyle's law, right? Boyle's law, the size of the, the volume and pressure deal, right? When volume goes up, pressure goes down, vice versa. And then well, that comes up later, so I'm going to just skip that. All right. Differentiate between the following nerves: phrenic, vagus, glossopharyngeal in terms of number, type of muscle it innervates, and function. So, what's the name of the nerve that goes to the diaphragm? That's the phrenic nerve. Anyone remember the memory trick for what spinal nerves give rise to the phrenic?
C3, C4,
C3, C4, C5, keep you alive. I know that's a dumb memory trick, but if you ever go into neurology and you have to deal with spinal cord injury patients, remember that memory trick, okay? Makes the difference where there's, with a high spinal cord injury, those patients have to be on a ventilator because they can't breathe on their own, whereas with a lower spinal cord injury, they may not be able to walk, but they could probably breathe on their own, so it's something to bear in mind. All right, let's talk about the vagus and the glossopharyngeal. vagus is cranial nerve number 10. Glossopharyngeal is cranial nerve number
11.
What's the correct answer?
9.
It's 9. Okay, good. It's 9. Both of them are branches of the parasympathetic division, so remember that. Glossopharyngeal and vagus are both part of that. So let's talk about let's talk about the vagus first. So the vagus is really important for modifying the heart rate. It connects up with heart muscle. What does the vagus nerve do to your heart rate? Speed it up or slow it down?
Slow it down.
Slow it down. All right, it's part of the parasympathetic division, so it's going to slow down your heart rate. Okay. The vagus also does something for your lungs that's pretty important. It monitors how stretched the lungs are. Okay, to prevent like overstretching when you inhale. There's that thing called the Herring-Brewer reflex, and it's the vagus that's monitoring that stretch information. Now, both the vagus and the glossopharyngeal are important for monitoring your blood. They both monitor the pH of your blood. They're hooked up to what are called chemoreceptors that are in the walls of your blood vessels that monitor the pH of your blood. The chemoreceptors also measure carbon dioxide levels in the blood. So for example, if the vagus and the glossopharyngeal detect that your pH is low, and detect that your CO2 levels are high, they will then send that information to the respiratory center, and that will then probably make you breathe faster and deeper to try to get rid of that CO2 to try to bring your pH up, okay? So they're really important for sort of controlling how fast you breathe by monitoring pH and CO2 levels in the blood. In addition, they're also connected to baroreceptors that are in the walls of your blood vessels. What do baroreceptors monitor? Pressure. So the vagus and the glossopharyngeal can monitor pressure as well as those chemicals like the pH and the CO2. So lots of important stuff. All right, what percentage of CO2 travels in the plasma and what percent is bound to hemoglobin? So first of all, there were 3 numbers here to memorize. What's the percentage of CO2 that's just dissolved in the plasma? 7%. What's the percent that's attached to hemoglobin? 23%. And by the way, when carbon dioxide is attached to hemoglobin, you call it carboxyhemoglobin, versus when oxygen's attached, you call it oxyhemoglobin. Anyway, so 7% is dissolved in the plasma, 23% is carried attached to hemoglobin. That then leaves 70% that's carried, or that's transported in what form?
Bicarbonate.
Bicarbonate, okay? So most of the CO2 in the blood, just to remind you, is transported in the form of bicarbonate. And we'll talk about that in a moment when we talk about the chloride shift. Keep in mind that it's the part of CO2 that's dissolved in the plasma that gives you what's called your partial pressure of CO2, or your PCO2. That's a value that's around 35 to 45 millimeters of mercury. So PCO2 levels in the blood, 35 to 45 millimeters of mercury, whereas PO2 levels, which are due to how much oxygen is dissolved in the plasma, are usually around 100 millimeters of mercury, okay? Again, in the case of oxygen, roughly 1.5% of oxygen is dissolved in the plasma. That gives you the partial pressure of oxygen. And then 98.5% is attached to hemoglobin, and that gives you the oxyhemoglobin. Describe internal respiration, chloride shift, and the Bohr effect. So first of all, what's internal respiration? That's gas exchange between what and what? The capillaries and your tissues, right? What were the other names for, there were two other names for internal respiration. We also call it systemic respiration, or we also call it tissue respiration. What's the type of respiration called that's going on in the lungs?
External.
That's external respiration, or it's also called pulmonary respiration, right? All right, the chloride shift. So the chloride shift is a little move that exchanges chloride with bicarbonate, and it's the reason why you're able to carry CO2 in your blood in the form of bicarbonate. And basically what happens is, as CO2 is given off from your cells as waste products, again, remember, when you do cellular respiration, you're going to generate lots of CO2. We covered that in chapter 25. That CO2 gets into your blood, and then it crosses the wall of the red blood cell. It undergoes a chemical reaction where it's combined with water. You temporarily get carbonic acid, but then that breaks apart and you get hydrogen and bicarbonate. This is inside of the red blood cell. The bicarbonate, though, doesn't stay inside of the red blood cell. Instead, it goes through the wall of the red blood cell into the plasma in exchange for chloride coming from the plasma into the red blood cell, and that's what's known as the chloride shift. What's the Bohr effect? Is that what the effect that I have on students when I lecture? No. That's when you shift the curve to the right, the oxygen hemoglobin dissociation curve, and what specifically causes that curve to shift to the right? A low pH. So the Bohr effect specifically refers to a shift in the curve to the right due to a low pH. And remember, when the curve shifts to the right, it means that hemoglobin is letting go of oxygen more readily so that it can go out of the red blood cell into the plasma and then cross the wall of the capillary to get into the tissues so the tissues can have oxygen. Okay? And again, it sort of makes sense if you think about it, right? When a cell doesn't have enough oxygen, it's going to start generating lots of what? Well, let me ask you this. What becomes of the pyruvic acid at the end of glycolysis? So if you don't have enough oxygen in a tissue, it's going to generate lactic acid. That'll bring the pH down. That'll trigger the Bohr effect. And it's kind of like the red blood cells are helping out. They're like, okay, tissue, you've clearly proven that you're starting to run out of oxygen. Let me help you out by letting go of the oxygen to help, kind of meet your needs. Okay? That's at least the way I look at it. If you don't want to look at it that way, if you just want to memorize it, knock your socks off. But knock yourself out. Don't knock your socks off. Just knock yourself out. Wrong saying. All right. What area of the brain is responsible for regulating and coordinating breathing? The pons and also the medulla. All right. The basic respiratory centers are located in the medulla, but then there's centers in the pons that, like the apneustic center and the pneumataxic center, that adjust that. All right. Finally, we're in the digestive system. says, describe the organs of the digestive system in terms of tissues present, divisions, and functions. Fine. So let's start off. Where is the first part of your digestive system? Your mouth. Perfect. What's the name of the white things that are in your mouth? Teeth. Awesome. We're off to a good start. And you start there by eating and chewing your food. Fine. Where does the food go after your mouth? Goes into your pharynx. Yeah, specifically oropharynx, right? After the pharynx, where does it go? Into the esophagus, right? Esophagus is basically just a food tube. Then after the esophagus, you get into the stomach. Now there's a fair amount to know about the stomach. It's basically a storage area for the food. It's a reservoir. Some digestion, of course, is going on in there. And there's a lot of different cells that make different things. For example, what's the name of the cells that make hydrochloric acid and intrinsic factor? Those are parietal cells. What's the name of the cells that make pepsinogen and gastric lipase? Those are the chief cells. What's the name of the cells that make gastrin?
G cells.
G cells. Good. The ones that make mucus? Well, there are goblet cells, but then there's also surface mucus cells, and there's mucus neck cells that do it as well. So that's the stomach. All right. Other thing that's special about the stomach too, how many layers of muscle are in the muscularis layer of the stomach?
3.
3. Remember, that's kind of the exception to the rule, right? Most other places have how many layers?
2.
2, right? So most other places you have a inner what layer?
Circular.
Circular, and an outer longitudinal. But with the stomach, you also have an inner oblique, then you have the circular, then you have the outer longitudinal, right? And that's just, it kind of helps the stomach to be able to contract in all different kinds of directions to really churn up your food. All right. Next, what comes after the stomach?
Small
The
intestine.
duodenum, or the small intestine, specifically the duodenum, right? And the duodenum, just remember, or the small intestine in general, just remember that when you're in the duodenum, there is still a heck of a lot of digestion happening there, okay? There's probably more digestion happening in the duodenum than there is in the stomach itself, and that's because of all of the digestive enzymes that are being put into the duodenum from either the pancreas or from like the liver, right? But as you progress along the small intestine and get into the jejunum and the ileum, it shifts and it becomes more about absorption at that point, right? Anyway, so back to the duodenum. You got the pancreas emptying into there. Let's talk about the pancreas. Pancreas, usually the first sentence of every chapter I've ever seen, or every paragraph about the pancreas, usually starts off something like this. The pancreas is both a blank and a blank. Anyone know? Thank you. It's both an exocrine and an endocrine gland. Histologically, tissue-wise, which is it more of, exocrine or endocrine? Exocrine, right? Remember, it's like 98.5% acini. They're the guys that make the digestive enzymes and all that other jazz. It's only like 1.5% what? Yeah, insulin and so it's the pancreatic islets, right, that make all the hormones. So just bear that in mind. Some of the enzymes. What are some of the enzymes that are made by the acini? What's the enzyme that breaks down starch? Amylase. What's the enzyme that breaks down fat?
Lipase.
Lipase. And then there's a whole bunch of enzymes that break down proteins like trypsinogen or chymotrypsin or elastase. So there's a bunch of them going on there. Now, hold on, does that come up later? Now, just because the pancreas releases lipase, which could, in theory, break down fat, what else do you need to help break down the fats? You need bile from the liver and the gallbladder to basically do what to the fat first? Emulsify it, which just means to sort of trick the fat into mixing with the watery secretions that are in your GI tract. Once you have that, then the lipase can really get to work, okay? Let's see, other stuff that dumps into the duodenum is obviously the common bile duct. True or false, the gallbladder makes bile? False. The liver makes bile, the gallbladder
Stores
stores
it.
bile, right? Can you live without your gallbladder? Yes. Can you live without your liver?
No.
No.
All right. Moving right along. Jejunum and ileum then, it's becoming more about absorption. What were the three things that helped increase the absorptive surface of the small intestine? So microvilli, and then just villi, and then one more thing. There's something on the inside, they kind of look like rugae, but they're not rugae. They're called the circular folds, or they're also called the plica circularis, okay? So, it's a real thing. I see skeptical looks. It's in the PowerPoint, so you know it's true. If it's in the PowerPoint, it has to be true, okay? If it's in print, it has
No,
to
that's
be
not
true.
true because we had that one slide in the blood vessels that said that type A personalities cause. That's a fallacy.
That's a fallacy, okay, all right. I feel like you're walking proof of that, but yet you say it's a fallacy. All right, here we go. Where am I?
It'd be like
All
that.
discombobulated here. Okay, so those are the things that help increase the absorptive surface: the plica circularis, the villi, the microvilli. Now, when you do absorption in the small intestine, what type of vessels absorb like sugars and fats? What type of vessels are we talking about? I'll give you two choices.
Yes.
Blood capillaries or lymphatic capillaries for sugars and sugars and amino acids. Blood. Did I ask that
You
question
said
the wrong way?
sugars and fats.
Blood.
I apologize. I apologize. I'm sorry. I thought I did because there's a lot of smart people that just did. I was an idiot. All right. So the blood. The blood is going to pick up the sugars and the amino acids, and then who picks up the fats? Yeah, the lymphatic capillaries. And what's the name of those lymphatic capillaries? Lacteals. And what is the form that they absorb those fats in? Chylomicrons. All right.
Wow, I think I know.
All right. Then after the ileum, you go through the ileocecal valve, and then you're in the large intestine. What's the first part of the large intestine called that's kind of dilated?
Cecum.
The cecum. Okay. Now remember, there's going to be some absorption going on in the large intestine as well. A lot of absorption of water happens there, which is important for like preventing too much water loss in your stool. Okay. What's connected sort of to the inferior and posterior part of it? The appendix, right, is connected there. Where do you go after the cecum?
Ascending colon.
Yeah, ascending colon. What's the name of that turn?
Hepatic flexure.
Hepatic flexure, or also known as the right colic flexure. Then you go across and you're in the transverse colon. Then you make another turn and go down. That's the that's the splenic flexure or the left colic flexure. Then you go down and you're in the descending colon. Then you go into that squiggly part that is like the sigmoid colon. Then you're in the rectum. Then you're in the anal canal, and then you're like outside the body. All right. Okay. Where was that? Good. Which dessert? Excuse me. Which
Wait, what?
dessert? I guess I'm thinking of those gummy things.
Are they good?
Which dessert? By the way, they're all gone now. I won't tell you what percentage of people ate what or anything like that, but they are all gone now. Okay. Which division of the nervous system promotes digestion? That's pretty. Yeah, parasympathetic. What's the nickname for the parasympathetic? Rest and digest. So if you remember that, hopefully remember that. What is the function of GIP? Well, this is more complicated. First of all, what was the first abbreviation, or what was the first thing that GIP stood for?
Gastric.
Gastric.
Gastric inhibition peptides.
Yep, gastric inhibitory peptide. And what did it do? What was the purpose of GIP when it had that meaning? Well, what does gastric mean? It means stomach. What does inhibit mean?
It's like stop.
It means to like, no, inhibit doesn't mean allow.
Opposite.
It means to like stop it from doing something, right?
Right? No, that's what prohibit means.
And so does inhibit. There's words that we have that can mean the same thing. They're called synonyms. Yeah.
No, but like,
He's the one that doesn't like
He's an English
synonyms.
professor.
Yeah. No, I'm sure there's a distinction, but they both mean to not allow something to happen. Yeah. Okay. No, you're right. You're detail oriented. I like that. No, but so it's inhibiting the stomach, right? It's a way for the duodenum to basically say, listen, I got a lot on my plate, I'm pretty busy right now, stop squirting more of your chyme in here, I'm too busy right now, I'm already stressed out, just quit it. And so like, the stomach will stop squeezing so much, it'll tighten up the pyloric sphincter, so it's not releasing as much workload into the duodenum, right? That's number one, when it was called, yeah. What does GIP also stand for?
Oh,
Yeah.
gastric.
No.
dependency,
Glucose,
Dependent inhibition.
no. Okay, I'll just tell you. Glucose-dependent insulinotropic peptide. It's kind of a long, you know, those jerks, they just wanted to keep, they were so set on keeping the same abbreviation that they had to come up with that crazy long thing. But basically, it's fairly simple what it does. What does it do in that context? Anyone remember? Anyone remember what it does? It has to do with when you eat a sugary meal. Yeah, so when a sugary meal enters into the duodenum releases GIP, and under that meaning, it's going to go to the pancreas, cause the pancreas to start releasing insulin. It's kind of like telling the pancreas, oh my God, I just ate a bunch of sugar. In a little bit, blood sugar levels are going to be really high. You might want to start putting out insulin so you can bring down those blood glucose levels. Okay? All right. Where are we? Okay. Okay, fine. Good. Metabolism and nutrition. I'm going to skip that. No, I'm going to skip that because I firmly believe in studying in the same order that things were presented. So let me skip that. That'll be the last chapter we cover. Now I'll go on to the urinary system, chapter 26. Okay. Matching category with the diagram of the nephron. So that's covered in this next section that says differentiate the parts of the nephron. This is the only place on the final exam where there is actually a picture. Okay. Don't flip out. Is it colored? No, it's black and white. Would you like a colored copy?
Yes.
Too bad. You're not going to get it. We don't have. I don't think we have. Do we even have color copiers at NCC?
Can't afford that.
We do?
No.
No, we don't. Oh well, listen. I'm not allowed access. That's for the VIP tutors. I don't get to go in there. All right. So here's how this is going to play out. Wait, did I already drew a really crappy. Oh, good. I erased it. I realized this morning that I've never actually drawn a nephron, and it looked awful. But anyway, here's how this is going to work. So let's suppose that, say, questions 43 through 46 are this little section here. So it'll be something like this. Don't ask me why I picked 43 through 46. I'm just giving that as an example. And then over here on the side, this is where you'll have your dumb little diagram that'll be in black and white of a nephron. So let me see if I can do any better than I did this morning with my crappy drawing. Looking about as crappy as I did this morning.
You're watching your contract.
It's a little bit better. Okay, hold
What do you
on.
mean by that?
I want you to do the job right.
All right.
I still remember last semester they cost you a couple of G's.
It should cost me a couple of G's.
I remember that.
You're spying.
No, it's fine.
Right, and
It's
then
obvious if I see what arrow.
So-
Okay. Watching.
all right, that is, you don't know how bad it was this morning. That's 100% better than it was this morning. All right, and then what they'll have on this little diagram is they'll have little letters.
Yeah.
So it's bonus.
Okay? And then over here, they'll have something like, this is the place where filtration happens. And you'll have to choose the spot where filtration happens. All right? Understand what's going on there? So let me run through this. What's that?
I'm complaining.
Oh, you're complaining? That's all right.
I know.
Make yourself jump.
There is, I should hook you up with this person that's in my A&P 1 class. She is the best complainer. You know what I'm talking about, right? That's in the front. Yeah, who's
Yeah,
this?
Emmy.
Yeah, she's like, she makes it an art form. You're good, don't get me wrong. But she's like, she's at another level. Yeah. All right, where was I? So let's talk about then the parts here. So I mentioned filtration. Where does the filtration step happen? Well, first of all, wait. There's 3 steps to making urine, right? The first step is always filtration. What are the other 2 steps? Reabsorption and secretion. Where on the nephron does filtration happen?
In the glomerulus.
At the glomerulus, right? At the renal corpuscle of the glomerulus. That's where filtration happens. Remember you have, what's the name of that capsule that surrounds the glomerulus? Bowman's capsule. Remember there's 2 layers to it, the visceral layer, the parietal layer. What was the name of the cells that make up the visceral layer? Podocytes, right? You had those guys there. So that's That's where the filtration step happens. Then obviously, anatomically, the next part you'd go into is the PCT. What's the PCT kind of famous for? What process? Reabsorption. Okay. Of all of the filtrate that you make in the first step, what is the total amount that you're going to reabsorb? 99% ultimately is going to get reabsorbed. And what fraction of that, or what percentage of that happens in PCT? 2/3 or like 66 or 65%. Okay? So it's famous for reabsorption. And what kind of modification do the cells that line the PCT have to allow them to be better at reabsorption?
Huh?
They have microvilli, right? By having those microvilli, it creates more surface area for reabsorption. Kind of in the same way that you got microvilli in the small intestine, it's increasing surface area to give you better absorption going on, or reabsorption, whatever you want to call it. All right, next you come to the loop of Henle. Note that when you're in the descending limb, it's usually relatively thin, and that means the descending limb is permeable to water. When you go into the ascending limb, it's oftentimes thick, and then it'll be not permeable to water where it's thick like that. After the loop of Henle, you then go into the what? DCT. And then after the DCT, you go into the collecting duct. All right. Remember that aldosterone and ADH, those are the areas where they like to act. They act on the DCT, they act on the collecting duct, etc. So if you measure, by the time you get to the end of the collecting duct, you basically have your final form of urine, and then it'll just drip out through one of those renal papillae. All right. Which division of the nervous system promotes urination? Parasympathetic promotes urination. Basically what it does is it relaxes the internal urethral sphincter, which is made out of smooth muscle. And then it also stimulates, what's the name of that muscle that's in the wall of the bladder? The detrusor muscle. So it's like doing two things. It's squeezing the muscle that contains the that contains the urine, right? And then it's relaxing the muscle that's kind of like the outlet for it. The only other thing to be aware of, though, is you also got that other sphincter called the external urethral sphincter, but that's made out of skeletal muscle, and that's why urination is under your voluntary control, right? Because you're controlling the skeletal muscle part. Differentiate between cortical nephrons and juxtamedullary nephrons. I'm not sure, is that typo still on there where it says juxtaglomerular nephrons? Or did they fix that?
Yes, that's
Yeah,
on there.
that's wrong. That, again, I can't change this stupid thing because I don't have the hard copy, the original hard copy or whatever, but anyway, that should say juxtamedullary nephrons. So, first of all, which do you have more of, cortical or juxtamedullary? Yeah, you have more cortical, about 85%, and then 15% juxtamedullary. Which are the guys that have vasa recta? Juxtamedullary, and that's because they have to supply blood to these really long loops of Henle that go deep into the medulla. Which of the two guys is more important for your ability to make concentrated urine? Also the juxtamedullary ones because of where they go. Speaking of making concentrated urine, what's the hormone that controls whether your urine is concentrated or not? ADH, good. Last it says, describe the process of urine formation and the countercurrent mechanism. So urine formation is basically those three steps. Always the filtration first, and then the combination of the reabsorption and the secretion that's happening later on. The countercurrent mechanisms, countercurrent exchange and all that stuff, that's all basically about creating a high solute level in the interstitial space of the medulla that'll create a drawing osmotic force that will try to pull water out of your tubules. Again, water will only be able to be pulled out if you have aquaporins actually in place, but that's what's creating the driving force to try to basically pull it out. God, I'm glad I don't have to take this. All right, we're moving along. Fluid electrolyte, acid-base homeostasis. So it says matching category with the following choices: sodium, potassium, calcium chloride, PO4 -3, by the way, is phosphate, in case you were scratching your head. So that's covered in this next section. So where do you find sodium in the body? Extracellular. It is the major extracellular cation. Okay. What's the major extracellular anion? Chloride. And the two, chloride usually follows the sodium around. So they're both extracellular. Why is there so much sodium in the extracellular space? Because of the sodium-potassium pump that's constantly working 24/7. It's pretty much on all body cells, and it's putting high amounts of sodium outside of your cells. Sodium is important for electrical signaling in neurons. Don't ask me why, but they kind of threw in a little detail about that electrical signaling, which I'm going to have to remind you about. When you're doing electrical signaling in neurons, you may recall there were things called graded potentials and action potentials. Sodium is an ion that usually diffuses into a neuron during depolarization, okay? Depolarization is when the membrane potential is getting less negative and going closer to zero, okay? The other major ion that's involved in electrical signaling in neurons is potassium. Potassium, though, is usually found where? Inside or outside of cells?
Inside of
It's inside of cells, so it's the major intracellular cation for the same reason that sodium was outside of cells, and that again is the sodium-potassium pump that's constantly putting high amounts of potassium inside of your body cells. Now, in terms of electrical signaling, it's potassium that diffuses out of the neuron during the repolarization step of an action potential, okay? Again, I have no idea why they put these details on there. As far as I'm concerned, this is like A&P1 information, but whatever. Calcium, I love calcium. Calcium makes your bones hard, but what else is it important for? Makes muscles contract. What else is it important for? Yeah, neuron transmission.
Yeah.
It's literally calcium rushing into the ends of the neurons that causes exocytosis of those neurotransmitters into the synaptic cleft. Okay? It really, I didn't belabor this point, but it's really important for exocytosis of all kinds, not just the release of neurotransmitters, so it's important for that. Calcium is also important to turn on enzymes. Oftentimes calcium has to attach to an enzyme to really turn it on, and that happens, for example, in the coagulation reactions. All right, there's a couple steps there where enzymes need calcium around to turn them on to make the next step happen. And then lastly, phosphate. Phosphate, where do you find, well, how many phosphates do you find in ATP? 3 phosphates. Phosphate is also a super important component of your nucleic acids. What do I mean by your nucleic acids? What are your 2 types of nucleic acids in your body? DNA and RNA, right? Again, this feels like A&P1 information, but it does come up. When you look at like your crappy model like this of DNA, remember there's this thing around the outside, and this is referred to as the sugar phosphate backbone. Okay? In DNA, the sugar is deoxyribose, and that alternates with phosphate. So it's like deoxyribose, phosphate, deoxyribose, phosphate, deoxyribose. So the message is phosphate is an important component of your DNA, and similarly, it's also an important component of your RNA. There's a lot of quirks on these final, like just little details that I feel like shouldn't be here, but I don't know, they pay me a lot, I don't want to argue. Just a lot of stuff, yeah. Not really. I like to say they do, but you know, they don't. All right, where was I
You sound
going?
like the type of guy you could put a price on your character.
What's that? Yeah, you can't, yeah, I'm not for sale. Screw those guys.
Except Black Kevin.
Getting all mad now. My blood's boiling. Okay, matching, so wait, where are we? Matching category with the following choices. Respiratory acidosis, respiratory alkalosis. That's covered in this next section where it says differentiate between the 4 acid-base imbalances in terms of causes, compensations, levels of pH, bicarbonate, and carbon dioxide. So fine. If you've got a decreased blood pH and an increased CO2 level, what problem is that?
Respiratory acidosis.
That's a respiratory acidosis, and it's often due to not what? Not breathing fast enough or deep enough. So it's due to hypoventilation. Okay? And if you have an increased blood pH and a decreased CO2, that's called what? That's respiratory alkalosis. Okay? Increased pH, decreased CO2 is respiratory alkalosis. And what's the cause of that? Hyperventilating. You're breathing too quickly or too deeply. Then we have the other issues. What's it called if you have a decreased blood pH and a decreased bicarbonate level? That's a metabolic acidosis, and that can be caused by like losing extra bicarbonate in the kidneys, for example. And then if you have an increased blood pH and an increased bicarbonate, what's that called? That's a metabolic alkalosis, and that, among other things, can be due to excessive vomiting. So please only do moderate amounts of vomiting. Keep your vomiting in check. No excessive vomiting, please. Okay. What's that?
No, listen.
If you do any excessive vomiting, will I give extra points? If you do projectile vomiting, that might be kind of cool. I'd like to see that. It'd be like a fountain.
During the finals, you get an
Huh?
A? During the finals?
You are shameless, but I don't know. I'm getting pretty desperate at this point too myself. Maybe I'll allow it. Yeah, we'll see. Only can we put it up on TikTok and in the web and all that stuff.
Promises she'll do it.
Yeah, I can see the headline now, like desperate student, like throws away dignity for A, like something to that effect. Yeah. Now, let me ask you a different question. If you had a metabolic alkalosis, what might your lungs do to compensate? If you had a metabolic alkalosis, what would your lungs do to compensate?
Oh, inhale more CO2?
Well, there's a different way of saying that, but you would breathe less, you would hypoventilate, and that would cause the CO2 to build up in your blood, and then that would help to raise your blood pH. Yeah. So you'd hypoventilate kind of as a fix for a metabolic alkalosis. All right. Matching category, see, this was already repeated on the previous page. Differentiate between the four above, that was already covered too. There's a lot of crappiness here. Describe buffers. So what are buffers? They act fast to do what?
Balance the pH.
Yeah, to balance the pH so that you don't have big changes, so you don't fall outside the normal range, okay? And they're usually a combination of two chemicals. One chemical acts like a weak acid, the other one acts like a weak base. We covered 3 buffer systems in class: the bicarbonate carbonic acid buffer system, something called the protein buffer system, and then something called the phosphate buffer system. For whatever reason, the final exam hones in on the phosphate buffer system. I personally don't think it's the most important one, but fine, they hone in on it. And what you need to know about the phosphate buffer system is you need to know the 2 parts that make up the phosphate buffer. In other words, what's the chemical that acts like a weak acid? What's the chemical that acts like a weak base? And the answer to that is, did I leave that up here? No, I erased this. So in the phosphate buffer system, the chemical that acts like a weak acid is dihydrogen phosphate. And that's going to look like that's going to look like this. So it's going to be H2PO4 minus 1, and so that's the weak acid. The weak base is monohydrogen phosphate. Let's see here. And that's going to, as you might guess for you chemistry people, it's going to look like this. Okay? So that's going to be, I got a squeaky thing here. It's going to be a weak base. Okay. So there you go. That's the phosphate buffer system. That's the parts that make it up. All right. Now we're going to skip back and go to 25 again, since we already did reproductive and development inheritance. So back to 25. It says matching category at the following choices: glycolysis, transition, Krebs cycle, etc. None of these. By the way, I believe on the final exam they did update it, and I think they actually removed that transition step as a possible answer choice. Okay. However, when I do the review now, I'm going to still mention some things about that. Okay. Just so you have the full picture. So let's go through this then. So first of all, glycolysis. Where in the cell does glycolysis happen? Cytoplasm. What's the starting chemical with glycolysis? Glucose. What are the ending chemicals you get? Yeah, you get pyruvic acid, or pyruvate, 2 of them, right? Do you need oxygen for glycolysis? No, it's anaerobic. How many ATP do you make directly from like 2 ATP directly, but then you also make some what that can be used later? NADH, right? That electron carrier that can be used later. What becomes of the pyruvic acid if there is no oxygen around? It gets converted into lactic acid. By the way, that process of converting the pyruvic acid into lactic acid with no oxygen around is referred to as fermentation. I know I didn't use that word. Fine, there it is. Now I've used it. Okay, so fermentation is the name for that. But let's suppose that there is oxygen around. What becomes of the pyruvic acid if there is oxygen around? It goes through the transition reaction and gets converted into acetyl-CoA. Okay. All right. Then the acetyl-CoA can enter into what?
Krebs cycle.
The Krebs cycle, also known as the
Citric acid
citric
cycle.
acid cycle, also known as the
Something I don't want to say.
tricarboxylic acid cycle. So you didn't know that one? I think I'm going to convert it all over to that now since no one knows
Yeah,
that.
you made that joke. I just heard it in the lecture.
That's
why
such
I-
a bad form. You know what? No one's supposed to call me out of my repeat jokes. You know,
I just heard it. That's
like
why.
97% of my jokes are repeat jokes, but you're not supposed to know that.
Intrusive thoughts about it again.
God, you're killing my jam, Jess. I'm sorry. All right. Where am I going here? Shoot. Listen, if you guys have any new joke material, submit it, okay? I will give you credit. I will say I will not pass it off as my own. I will give you credit. It might freshen things up a
He'll
bit.
at me and then
I'm so angry. I don't even know where I was. I lost my train of thought. Krebs cycle. Okay, Krebs cycle. So where in the cell does the Krebs cycle happen? In the matrix of the mitochondria, right? Does it require oxygen? Yes, it requires oxygen. How many ATP do you get directly from it, assuming that you started with 1 glucose? You get 2, right? But you also get some NADH, and then what's similar to NADH? FADH2 that can be used later on. Okay. And then what's the last step then? The electron transport chain. Where in the cell does that happen? On the inner mitochondrial membrane of the cristae. That's a good answer too. What's the name of those proteins that are embedded there? Cytochromes, good. Does it require oxygen?
Yes.
Absolutely. In fact, it's really that final sort of acceptor of the electrons and the hydrogens, and when oxygen does that, it becomes what? Metabolic water. How much per day? 200 milliliters per day. Not a drop more, okay? All right, and then how much ATP do you make from that process? 32 or 34. When you make ATP during that process, what's that method of making ATP called? Oxidative phosphorylation, versus when you made it during those other steps, it was called substrate level phosphorylation. All right, good. That's it. Okay, my God, we're at the end here. Any questions about any of that stuff? Awesome.
Fun activity.
All right, fun activity. Okay, wait, you gotta remember, you're training to